Okay, so this is the answers for the Double Award Biology Foundation Tier 2019 paper. So the first question says, the diagram shows a plant called camel thorn. Take the genus name for the camel thorn. So when we are looking, we know that in the brackets, it's told us the scientific name, which is here. And then we need to remember that the genus is the first one, so it's the Alhalgi. Camel thorn lives in dry desert areas where there are herbivores. Choose two labelled features shown in the diagram to explain how each is an adaptation for camel thorn to survive in deserts. So we've got small leaves as it reduces heat loss, and we've got sharp spines to protect from predators. Megan investigated growth in camel thorn plants in two different conditions. Plant A was given 250 centimetre cubed of water each day. Plant B was watered once a day at the start of the investigation. The diagram shows the appearance of the two plants at 7 days. Compare the root growth of the two plants and suggest how root growth in plant B is an adaptation to dry soil. So plant A, the roots are wider, which you can see they're spread out wider and they're close to the surface of the soil. Plant B, then the roots are narrower and they go deeper underground, and the reason that is anything to find water from the desert. The poster is about some uses of stem cells. So we've got muscle cells, blood cells, nerve cells, cardiac cells, liver cells and intestinal cells. Stem cells can differentiate. Using one example from the poster, explain the meaning of the term differentiate. So stem cells that can turn into specialised cells, because these are all examples of specialised cells. And a specialised cell is a cell adapted for a specific function. Stem cells undergo rapid mitosis. The table gives 5 features of cell division. Some features relate to mitosis and some relate to meiosis. Complete a table by writing true or false in each row to show the features that relate to only mitosis. So daughter cells that are genetically identical is true. Which is 4 daughter cells. It's false. In mitosis, we have 2 daughter cells. Daughter cells retain the original number of chromosomes. That's true because they're identical to the mother cell. Daughter cells have chromosomes in pairs. That's true. And it produces gametes. That's false. That's the function of meiosis. A patient receiving stem cell therapy may be treated with cells taken from their own body or from embryos. Give 3 advantages of the use of stem cells taken from a patient compared with those from an embryo. We haven't got to find a donor because we're taking it from their own body. There's going to be no rejection then because they're going to recognise the antigens, and there's no ethical issues of destroying potential life in the use of embryonic stem cells. The diagram shows a pathogen that causes an infectious disease. Say the type of pathogen that's shown above. Because it's got the protein coat here, we know it's a virus. Sudden changes in a gene causes changes in the antigens on the coat of the pathogen. State the term for a sudden change to a gene, that is a mutation. State one way by which pathogens are spread between people, it's direct contact. The following statements describe how the body responds when it's invaded by a pathogen. The statements are not in the correct order. Complete the sequence and put the statements in the correct order. So we start with number 2. Pathogens enter the blood. The pathogens have foreign antigens. Then we go to 5. The antigens are recognised as foreign by the lymphocytes. They respond by releasing antibodies to the antigens. The antibodies attach to the pathogens, number 4. And then lastly, the attached antibodies help phagocytes destroy the pathogen. The diagram shows a section through the skin to state the name of structures. So we have here, this is the sweat gland. We can't put duct or pore, it's the gland. And then Y is the capillary. We could also write blood vessel. Carbon investigated the control of body temperature. He set up a model to demonstrate the action of sweat using an apparatus shown below. At the start of the investigation, Carbon added 200 centimetre cubed of water at 80 degrees to each flask. He then recorded the temperature of the water in both flasks at 2-minute intervals for 10 minutes, and it's shown below. Complete the graph of the following results, plotting the point for flask B and using a ruler to join the points. Okay, so I've got 80, which I've got all down here. Okay. Then in Carbon's model, the towelling represents the skin surface. Use the results from flask B to explain the effect of sweat on human body temperature. So first thing, when we're warm, The sweat is released from the sweat pore. So the sweat gland produces sweat and the sweat is released from the sweat pore. It releases heat from the body because it evaporates as sweat. So as the sweat reaches the surface of our skin, it evaporates because of the heat from our body. With that then, our body temperature will decrease. Heat stroke is a condition in which body temperature becomes dangerously high. Use results from Flask A to conclude why people who do not drink enough water during very hot weather may suffer heat stroke. If you look at the temperature change, there's not a lot of temperature change. It remains quite high. So the first thing I want to say is that the temperature remains quite high, which we can see. So they're going to overheat, and as they overheat, less sweat is produced, and that's why they're overheating. The photograph shows an adult North Sea cod. Cod are important in maintaining biodiversity in the North Sea. Choose the letter A to C from the list which defines biodiversity from an area. And biodiversity is the number of organisms within the species. Read the following information about cod. Overfishing in the second half of the 20th century has had a serious effect on the stocks. The total stock of adult cod is estimated annually. The safe stock is the minimum mass of the adult cod population that should be left in the sea each year to ensure that the cod remains at sustainable levels. In 1996, scientists predicted that without action, there'd be no adult cod in the North Sea by 2015. In 2005, the European Commission put an annual limit in the mass of cod that could be taken by each vessel fishing in the area. The graph shows the annual catch of the cod and the stock of adult fish remaining in the North Sea from 1965 to 2015. The safe stock is 150,000 tonnes. Use a ruler to draw a straight line on the grid to show the safe stock from 1965 to 2015. So as you can see here, at 150,000 tonnes, I use a straight line and draw my ruler. So anything below that is below the safe stock, and above we've got loads of cod. Use the data in the graph to answer the following questions. Calculate the difference in mass between the total catch in 1982 and the safe stock. So if we go up to 1982, so 1, 2, we go up, go across and it's 400,000. We take away our safe stock, which is 150, which gives us 250,000 tonnes. 1 tonne contains 220 adult cod. Calculate the number of adult cod in the safe stock. So we're going to do 120 times the 150,000 tonnes, which gives us an answer of 18 million. Describe the evidence that shows overfishing beyond a sustainable level took place between 1971 and 2005 is because the numbers of cod decreased below the safe stock. Answer the evidence that supports the prediction made by scientists in 1996 that without action there would be no adult cod in the North Sea because the trend in stock will lead to extinction. Shows the fishing quota of 2005 has had an effect on the cod population is because the fish stock started to then increase after 2005. Maintaining biodiversity ensures a continued food supply for humans. Give one other benefit to humans of maintaining biodiversity. It's for medicines, to create new medicines, and for human well-being. Reaction time can be measured using the following method. The experiment holds a 30 centimetre ruler just above the hand of the subject. The ruler is dropped. The subject then catches the ruler as quickly as possible. The distance travelled by the ruler between steps 2 and 3 gives a measure of reaction time. You have 2 solutions of coffee, one with caffeine and one without. Describe how you would test the effect of drinking coffee with caffeine on the reaction time of a class of 20 Year 11 students. Include at least 2 variables which should be kept the same in order to make it a fair test. You are not required to write the steps 1 to 4 above in your answer. So we would test reaction time before drinking either of the coffees. You would give the person the decaffeinated coffee, and we do a blind test. We wouldn't tell them which one it was, so don't tell them that they're drinking decaffeinated or caffeinated. We would then give the caffeinated drink, carry out the reaction time test, measure the distance with a ruler to see how quick they caught it, and we would repeat the experiment to compare the results. Our variables, we make sure that they use the same hands, the same concentration of coffee, and the same drop height. State what is meant by the following terms: a gene, which is a section of DNA, an allele, which is a different form of the same gene. So it would only be big B, big B, big B, little b, or little b, little b. Okay, they're the three alleles that we have. Cystic fibrosis is an inherited condition. The allele that causes this condition is recessive. The family tree below shows how cystic fibrosis is passed on in families. So circle, unaffected female, square, unaffected male, this coloured in slightly affected male, and we've got an affected female. Use the letters N and N to complete the Punnett square to show how parents 1 and 2 could produce offspring number 4. So 1 and 2 are parents that are unaffected, but number 4 is a male who is affected. And just knowing straight away that if you've got a child who's got a recessive condition, they have to be little n, okay? And for them to both have little n's, both parents must have a little n because they pass on that recessive allele to their offspring. But because they're unaffected, they have to have a dominant allele, which then gives them the big N, little n. So as you can see here, I've got the heterozygous parents, and then we have the chance, and if I circle it, there's child number 4. Take the genotype of individual 9, explain your answer. Well, he's homozygous recessive, as little n, because he has cystic fibrosis, so he's going to need 2 recessive alleles for it to be in control. State the possible genotypes of individual 5. So individual 5 is a sibling. So individual 5 could be homozygous dominant, which you can see here, because it is a possibility, or they could be heterozygous. Our understanding of how characteristics are inherited comes from the principles proposed by Gregor Mendel in the 19th century. In his experiments on pea plants, Mendel studied characteristics controlled by a single gene. State why this is not represented by most phenotypes. It's because it's controlled by multiple genes. The diagram shows a bacterial cell. Label structures A, B, C on the diagram above. So this is stuff that they can ask from last year, labelling a plant, animal cell, etc. So we have the cell membrane, our cell wall on the outside, and then everything that surrounds is our cytoplasm. Karas wanted to investigate the effect of antibiotic on bacterial growth. She set up the experiment shown below with the test tubes containing increasing concentrations of an antibiotic and inoculated each test tube with a fixed volume of nutrient broth containing a known concentration of bacteria. The tubes were incubated at 25 degrees for 2 days. The cloudier the solution becomes, the more bacteria present. A clear sample means there is no bacterial growth. So we've got the tube number 1, 2, 3, 4, 5, 6 and the concentration of antibiotic. Explain the results observed in tubes 4 to 6. So 4 to 6, it's clear. So all the bacteria are killed and there's no bacterial growth. Suggest the minimum concentration of antibiotic that would be effective against the bacteria. You've got 1.5. Suggest how Karas could improve her experiments so that she could obtain a more accurate value for the minimum concentration. And it would be that we'd have test smaller intervals. We know it changes between 1 and 2, the concentration. So if we had tubes that had a concentration of 1.2, 1.4, 6, 8, and then 2, we can see exactly where there's a clear solution and there's no colour change.